Welcome to the Manhart Lab. And here we'll give a little glimpse into what we do in the Manhart Lab, all the instruments we have, and what our lab looks like, and the spaces that we all have access to, whether shared or our own, and what's available for new students. What's nice about our lab is we have a ton of space in here. As you can see, huge, nice, big lab space. Every student has their own lab bench right now, and there are some open lab benches for future students to come in, which is super nice. So you have kind of your own space and own shelves and drawers that you can use to kind of keep the stuff that you need to, keep your samples, keep all your stuff to prepare. Obviously our pipettes, which are our main tools in this biochemistry lab. But you have a lot of space in here to kind of make your own, which is super nice. Most biochem labs that need to grow cells and express certain proteins, we use our shaker. So we have a shaker in lab, which we use for all our cell growth and any transformations that we do. So you can see some flasks are shaking right now. So we have this in lab, which is super nice, and it allows us to have plenty of space to do. Basically, we can fit 6 liters of media in there pretty easily, and some other small flasks as well. If you need smaller tubes, you can put them in there as well. We have a minus 80 degree fridge, so that keeps a lot of our purified protein stocks and some of our other materials that need to be kept at minus 80 degrees. So vacuum sealed, so it's really nice to have this in lab, so we don't have to worry about keeping stuff at non-ideal temperatures, and can keep all our proteins good for a very long time. 4 degree fridge, so just a basic deli type fridge to keep anything that needs to be at 4 degrees C cool. We have our Teacan over here, which is just a very fancy spec, but allows us to do DNA and protein quantification to see how much DNA or protein we have in a given product. We can use it to monitor colorimetric assays, so any ATP hydrolysis experiments or anything else that uses colorimetric reagents in order to that are seen in specific wavelengths, we can actually use to test the optical densities using this instrument. We also have our own Milliq system, so if you need Milliq Water, we have that in lab, which is nice, super handy to have on hand. It allows us to, if we need a little bit cleaner water than just regular DI water, we can use this Milliq water, which is great for protein purifications and cell expressions and all of that. So here we have our sterile lab bench, where we can work with all our sterile lab materials. So anytime we need to do cell culture or anything that needs to be sterile, pouring plates, anything like that, selecting single colonies, streaking to single colonies, we can work here with all our sterile media and bottles up top there that we sterilize via our autoclave. And so below the sterile station, we have our incubators that allow us to grow cells on plates, and we can set them to, right now one's set to 37 degrees for E. coli, but we can set them to grow yeast as well. And also on this bench we have a photoreactor. So this allows us to do different photochemical cross-link reactions, where we have lamps of specific wavelengths that we can sort of, you can see, hit the reactions from all angles, and if it needs to be photoactivated or activated via a specific wavelength, whether it's cross-linking or some other experiment, we're capable of doing that. Here we have our radiation station, which allows us to work with our radioactive materials. So we use P32 in our lab to label DNA. We can also use it for ATPase or ATP hydrolysis experiments. So we have a safety area corded off different areas behind plexiglass, so we can safely work with our radioactive materials. And then if we want to run a gel with radioactive materials, you can see a gel running right now. This is our high voltage gel box that's running. It's a urea gel, so this allows us to run urea page gels to run denaturing experiments and in the hood with radioactive materials. And over here we have another lab bench area with instruments on it. So we have our PCR thermocycler for all our PCR experiments. We have a quench flow that we're in the process of trying to put back together. It's a little bit outdated, but we're trying to get it up and running. And then here is our gel imager. So this allows us to image any gels that we run in the lab if we're looking for fluorescence labels or radioactive labels of DNA. We can identify them on the gels using this scanner, which makes it super nice to have one in our lab so that we don't have to go anywhere to scan anything, and we can do it whenever we need to. And so it's scanning right now. You can see it working, and then it'll come up on this computer, and then it'll show you for this specific gel. This is a radioactive gel, so it'll show you the DNA that's labeled with P32 where it appeared on the gel. So here we have in the back of our lab, which is really nice, we have our workspaces, our personal workspaces or our benches, so that everybody is able to do whatever they need during the day, work at their desk, keep their stuff. There's plenty of open desks, which is great. Here's my desk, a little messy, but as you can see, lots of desks and the opportunity to come in and kind of set up shop and get all your work done that you need to that isn't lab work. So for protocols, data analysis, if you need to work on homework for your classes once you're in lab, you have that space to do so. And this is our shared instrument room. So this is shared with the Manhart Lab as well as some other biochemistry labs, and really anyone who needs any of the instruments in here can get access to this room. So we have our autoclave, which the Manhart Lab is in charge of. So anything you need to sterilize, liquid or dry sterilization, if you need to sterilize waste, you can use our autoclave in here. So this is very useful, one of our most useful equipment. And then we also use our nice temperature controlled centrifuge that we spin a lot at 4 degrees C. So when we're spinning down cell pellets to express or for purification to lyse cells, we can use the centrifuge here. So this is owned by the Stanley Lab, but we have access to it. We just put in our password and can use it. We have access to a sonicator as well, so if we need to lyse cells by sonication, we can use that right there, as well as another gel imager tool. And then we have one in our lab, so we don't need to use this, but in a pinch we can use this to read at specific wavelengths this gel box if we need to read agarose gels. And then some other fridge and centrifuge items that the Stanley Lab has that are also available for use if need be.